Malcolm, tell us, what are you doing here in Lisbon this week?
4 things.
Right.
One was to attend the Strati conference at the Gulbenkian Foundation. The second was to meet with other members of the International Commission on Stratigraphy. I also wanted to carry on a little bit of field work that's a continuation of previous work. And also it was an opportunity to meet a Brazilian co-worker that I've worked with for the last 20 years on the geology of eastern Brazil. And he now lives in Portugal, so it was the quickest way of meeting him.
Fantastic.
Obrigado por ter vindo. Malcolm Hart é professor emérito de micropaleontologia na Universidade de Plymouth. É por aqui. Okay. E obteve o seu grau em geologia e também o seu doutoramento na área de paleontologia em Cretáceo no Imperial College em Londres. Ele é autor de cerca de 200 artigos científicos, editor de 8 livros, editor-chefe da revista internacional Cretaceous Research. Também é presidente da Comissão de Geoconservação da Sociedade de Geologia Inglesa e foi membro do Conselho de Natureza do Reino Unido. O professor Hart faz parte do grupo consultivo para Ciência e Conservação do World Heritage Site do Dorset e Devon, e também da direção de English Riviera Geopark. Mas o Malcolm também tem um bom conhecimento da geologia costeira aqui em Portugal, e tendo começado a interessar-se por estes assuntos na década de 90, em colaboração com as universidades de Lisboa e Coimbra. Este trabalho de micropaleontologia e bioestratigrafia foi realizado desde Cabo Especial até Montejo. Então, nós estamos aproveitando este dia do professor Hart em Lisboa por ocasião da conferência do Strati e convidámo-lo a apresentar as suas mais recentes descobertas na área de acidificação do oceano. Thank you, Malcolm, for joining us, and it's over to you.
Thank you very much for the introduction. I should explain a little bit about what's on the screen. Some of you may recognize the logo for the European Project on Ocean Acidification, IPMA, and above that is our Marine Institute, which links the university with Plymouth Marine Laboratory, the Marine Biological Association, and also SAFOS, all of which are based in Plymouth and have links to the university. I've put at the bottom there, in collaboration with several people. Alba Sanchez is a Mexican marine biologist, and you'll see the connection shortly. Bruna Dias is, and that's terrible pronunciation, she's Brazilian, who came from Brazil to work with us, and we helped train her. Deborah Wall-Palmer is a postdoctoral fellow and a marine biologist who works with us in geosciences. Laura Pettit is a PhD student, also a marine biologist, and Jason Hall-Spencer and Rosa Maria are also both professors of marine biology. So what we're doing is trying to work with marine biologists as well as geologists and paleontologists to look at the issue of seawater or ocean acidification. I probably don't need to explain what the other carbon dioxide problem is. That is the concept, or actually the information, that the world's oceans are gradually becoming more acidic. And there are predictions as to how far this process will go, depending on the rate of carbon dioxide increase in the atmosphere. And most people are concerned about how that will impact on both the metabolism of marine organisms but also on their ability to calcify. But from our perspective as geologists, we always look at a modern event like ocean acidification and try to think, well, have we actually seen this before in the geological history of the planet? And if we have seen it before, how did it show itself, and what were the effects that are greater than those that we are seeing today? So that's where we're coming from. So that is simply just summarizing what I've said in terms of increasing PCO2. Experimental work in mesocosms and microcosms are absolutely fine, and we have cultures of foraminifera and other marine organisms in our laboratories. But the first example, and I'm going to use three main examples. The first one is the island of Ischia in the Mediterranean. And Ischia is in the Bay of Naples. But on the island itself, in the area with the red box, there is a small peninsula which has a whole series of gas vents. And these are carbon dioxide gas vents. Now, the most important thing is that these gas vents are turning the water acidic. As you can see along the southwest coast, the pH goes from 8.14 down to 6.57, whereas along this north coast it's 8.14 to 7.09. And that is simply caused by these carbon dioxide gas vents. Now, importantly, the gas is at normal temperature. In other vents in Indonesia and round Stromboli and Vulcano in the Mediterranean, the water temperature is being elevated to about 80 degrees centigrade. And therefore any biological response is probably more the result of temperature than actually the acidification. Ischia is important because it is at normal water temperatures. So, I hope you can see this. The light's a bit bright there, but and also the sound effects. I think I'm the only person who's going to get the sound effects. I can hear a lovely gurgling noise here, but you can see the gas bubbles of carbon dioxide coming into the area, just coming through the sediment, which is a calcareous sand. In some places, the increased PCO2 has created lush seagrass beds. And this is some of our marine biologists collecting gas samples, water samples. They're also collecting the fronds of the seagrass, the roots, and everything else that we can sample. Now, the marine biologists were looking at the mollusks, the echinoderms, and all the other organisms that are present within these samples. Now, we were looking particularly at the samples from this north coast and the south coast to give us a spectrum of levels of acidification. We were also looking at the benthic foraminifera that are associated with seagrass beds, and in particular species that attach to the roots, those that live epiphytally on the leaves. Now, the interesting thing to us was the fact that none of these foraminifera are deformed. In many environments, foraminifera grow deformed tests. And this now seems to be restricted to where there is metal contamination. pH does not seem to generate any deformation, and that was a surprise to us. Now, for those of you that aren't familiar with this sort of diagram, these are sort of standard ways that we illustrate micropaleo data. Professor John Murray many years ago devised this tiny little triangular model, and what happens is that as the normal environment of a Mediterranean seagrass is a mixture of these calcareous forms and this other group of calcareous taxa, as you look at a normal sample, they are exactly where you'd expect them. When we reduce the pH down to about 7.8, 7.9, it migrates to an assemblage that loses this group, and we have calcareous forms, but also beginning forms that were really so rare, less than 0.1% in the normal environment. These are forms that have a chitin lining, and they adhere sand grains to them. There may or may not be a small amount of calcareous cement. But then when you go to the much reduced pH, about 7.6, all you find, you've lost all these calcareous forms, and all you have are these forms with a chitin lining and these sand grains that are stuck by means of tiny chitinous threads to make a test. Showing this a different way, just in terms of diversity of species, the diversity drops to round about 1, 2, or 3 species of these so-called agglutinated taxa. And when we look at the percentage of calcareous forms, again, by the time we get down to 7.6, 7.5, there are no calcareous taxa present. Now, I'm not a biologist, so I'm treading on dangerous territory here, but when we look at the way in which foraminifera create their shells of calcareous material, and this is experimental work by Lennart de Nooijer in NIOS in the Netherlands. What Lennart has found in a series of papers, particularly in 2008, 2009, is that internally the foraminifera generate a pH between 8 and 9 to create their calcareous test. And that's internal. So if the foraminifera are generating an internal pH of 8 or 9, when the external pH is about 8.16, 8.17, then to raise the internal pH to 8 or 9 when the external pH is 7.5, 7.6, this means they're investing too much energy into doing that, and simply they can't survive. And so what we're finding is that this internal raising of pH to create or secrete the calcareous skeleton is just becoming impossible with the level of pH in the water outside. So the data from Ischia gives us an idea that in a really acidified environment of about 7.6, 7.5, we would find no calcified tests. We would only find those created by these agglutinated species. But of course, when you're looking back in geology, you have to make sure that what has happened to the sediments or the rocks after they were formed has not changed what you are seeing. And this is where the geologists have to put their training into perspective and say, well, what has happened to these sediments post-deposition? And are we seeing what was really there at the time they were alive? Various experimental work has been done, particularly by a group based in the Netherlands, Belgium, and also Italy. Nguyen et al. Published a paper on it, and they showed that down to 7.5, the calcareous forms disappeared. But they were particularly interested in this event in the Paleocene-Eocene thermal maximum, which is round about 55 million years ago. And it is thought to be the result of almost an immediate, geologically immediate release of carbon dioxide or methane into the environment. However, what happens is it leaves an isotope signal, and this is the isotope signal, and this carbon isotope excursion, it's known as a CIE, has been shown as an acidification event at the PETM. However, one problem I have, not with the results or the interpretation, but this appears to be formed by a sudden movement of the calcium carbonate compensation depth, because these are in deep water. And I argue with some people that a sudden movement of the carbonate compensation depth, while geologically very important, is that what we should be calling ocean acidification in the modern biological terminology. Yes, it's an acidification event 55 million years ago, but it's in very deep water, and it's a migration suddenly of the CCD and not an acidification event related to the surface. Because in shallow water environments, we find carbonate secreting foraminifera.